Thank you very much for the hoster to invite me to for talk here. I would like to talk about the project that we done in my postdoc actually. I finished it 2 or 3 years ago, but nowadays submission to a journal get published always take 2 or 3 years. Well, in our case, but it's luckily enough it's just published, and the title is single cell profile review novel memory B cell like subtype of follicular lymphoma with increased transformation risk. So I have nothing to disclose. So first, I'd like to give some background of this disease. So follicular lymphoma is type of cancer that originates in the B cells, so the B cell like immune cell cancer. So most frequent type of indolent lymphoma. So it's not gonna make your life dramatic at beginning, but 2 to 3% of patients transform to a more aggressive B cell lymphoma, mostly DLBCL, which is have very poor outcomes. So the treatment of follicular lymphoma depends on the disease stage, symptom, and patient's overall health. And all the treatment strategy will include mostly just watch and wait, and sometimes chemotherapy, radiation, transplantation, and there's some novel therapy, target therapy, and immune therapy as well. So you can see that the disease actually starts. Can we see? Okay, yeah, this disease starts from the germinal center of follicle of lymph node, and the cells expansion accumulate and eventually spread to other places of your body. So the knowledge gap here is really, the first is lack of knowledge of FL phenotypic subgroup and their biological characters. So especially single cell study for a large cohort, just to see how the immune profiling of the B cells or the tumor cells, as well as the infiltrating T cells and the other immune cells. And there's also unmet need for identifying patients with high risk of transformations. So there's some clinical tools we use, like the Flippy M7 Flippy, to predict a high risk patient, but the transformation risk is still something that we haven't achieved yet. So the goal of this study is to perform a deep immune phenotyping to characterize this tumoral heterogeneity, intra and intertumoral heterogeneity, and as well as infiltrating immune T cells. And again, I think we're talking about the CyTOF this morning, so we mainly use CyTOF. Sorry. So this is a study design. So in this study, we assembled a pretty large cohort, which included 155 follicular lymphoma patients and also plus 36 reactive lymph nodes, which is treated as normal control in our study. So all these 150 follicular lymphoma samples were collected in diagnosis without any treatment, just to have rule out any confounding factors. And so in BC cancer, when patients were diagnosed as FL using flow cytometry, usually we will collect the samples, excessive samples, and store in the biobanker. So as a preserved vial. So we want to take these vials for our study. So for our study, we have 2 cytol panels. Each one have around 40 markers. So one dedicated for B cells, so we have, and one dedicated for T cells. For T cells, we actually want to look at all these cytokines. So we actually stimulate the T cells and then stain and do the CyTOF later. So we also use some sequencing platform like single cell sequencing and target sequencing just to validate and confirm some of the finding in our study. So then basically a lot of time for the data analysis, pre-processing, normalizing, and also some Quokers analysis different expression. And we also have some clinical data that we can correlate with to see if there's anything interesting in that regard. So this is our CyTOF antibody panel. So 2 panels, as I mentioned. So B panel is mainly B cell activation, exhaustion markers, cytokines, all these kind of things. And also the T panel is also a marker related to different T cells in different subsets, and also look at the cytokines to assess the function. Before I show you guys the real data, so the one thing I really want to stress is like a QC is quality of the data that we're going to get. So like all these like big study and also require that like cover a long time period, like how to make data consistent is, and with minimum variation is a key for the successful of the study. So for this study, we actually assembled a pooled React lymph node. So which is like we put all like we collect the lymph node samples React lymph node samples from different patients and put them together and freeze them into many vials, and we run these vials same time as we run the samples in different time points. And then we use these samples just to assess where the data consistency between each run batch. So this is 17 batches of data we run for this study. So from 2017 to later 2018, and based on the dimension reduction process, all looks very similar to each other, just to be sure that we are collecting very consistent data. And there's also like quantitative measure. Here we use entropy. So every single data point, every single date we have two violin plot. The left hand is maximal, theoretical maximal entropy, which measure like how similar this pooled React lymph node is compared to the other React lymph node example we run in the other batch. So we can see that the observed entropy distribution is very close to the theoretical maximum, both for our B cell and T cell panel. Okay, so this is global mapping of B cells. So this is like a mapping of 192 patients, including 155 FL patients, plus 36 React lymph node patients. So there's actually, if you look at it closely, there's actually like 192 colors. So each dot represents one patient. I have to use some program to generate 192 different colors palette to just plot this plot. This is quite different from, the other study that we see. So the other study we are using is like we're looking at the DLBCL patient. DLBCL is an aggressive form of lymphoma. So when we look at the DLBCL, that each patient seems to like form a like isolated island. Like they're very different, heterogeneity. They're very different from each other. But FL, there's still like some kind of commonality. So if we plot them separate, like a React lymph node and versus FL, you actually see that there's some difference between these two groups. So React lymph node, they occupy comparatively narrow, phenotypic trajectory, while FL, like you see some cells like in this React lymph node area, but like a lot of cells are like actually spreading here, and there's also isolated island like the LBCL. Most of these islands is just comprised of like a single cells from single patient. So just to talk about the React lymph node first. So there's a couple, like we can see that there's some there are structures inside this island, and based on the canonical markers like the IgM, CD44, we can easily see actually the first half circle is naive memory B cells, and in the middle, there's likely to be memory B cell based on CD24 or CD27. And we can also see the GC, germinal-centered B cells, based on CD38, 40, 86, 20 HADR, these kind of markers. And there's also, you can see there's also some small subset of cells flowing on the top. They are plasmablast and plasma cells. So this is like when we try to plot these React lymph node cells individually for 36 patients, we can see the cells don't really move around, move out of this normal trajectory or normal boundary. But what the difference is, like a composition of these cells changed from sample to sample. So to measure the quantitatively, like we actually use a turn rate plot. So each dot here, it represents a patient, and the location of the point, location of the dots inside the triangle is actually telling the composition of naive GC and the memory cells. So you can see that in the reactor lymph node page example, there's a comparatively large amount of naive B cell, and around the 20 or like 15% of memory B cells, and also some around 10% of GC B cells. So they are like a very consistent composition in the reactor lymph node. But compared to follicular lymphoma, most of the follicular lymphoma is already done with germinal center B cell, indicating that maybe it's a disruption of GC structure. And the majority of the cell left over, normal residual cell, are naive, either naive or memory B cells. So we cannot really eyeballing like just to see like how what all this population is. So we actually perform a phenotypic clustering just to give us another idea, like unsupervised clustering, how many clusters we can have, and whether they can do a good job to separate the cluster, different cell population based on the immune phenotypes. So the phenotypic yield 78 cluster. 6 of them, I think, is from reactor lymph node. So we can see actually it's separated pretty well. We can see naive memory, two kinds of memory B cells. One is IgG positive, and one is IgG negative, mostly IgM positive memory cells. And there's also GC plasmablast and plasma cell. But for the FL side, it's really difficult to comprehend. It still have like 70 something clusters. So we decide to do another round of clustering. So usually we call the meta clustering, using hierarchical clustering based on the immediate value of expression pattern. So when we cluster these 78 phenotypic cluster using hierarchical clustering, we end up with 19 meta clusters. This is colored by, so you can see it actually colored on the top of the heat map, and also in this graph. It's combined the two memory cluster into one. So we don't really care. But here, like we can see that there's interesting things we see in the FL examples, which is like, so on this side, it's mostly resemble the germinal center B characters, like meta cluster A. It's a very big cluster, and also CFE with some like C is more like primary germinal center, and also the L is more like germinal center entry cell, so which is like a very late stage of germinal center. So this is really in contrast. Sorry to mention that. Forgot to mention that there's also one big branch that coming out from here, which is very close to naive and memory B cells. So this is very interesting observation, which is in contrast to the traditional classical belief that the FL is derived from germinal center B cells, because we see that some FL, or at least some subset of FLs, behave or phenotypically looking like memory or naive B cells. So we look at like mainly focus on two metaclass, which is metaclass A, metaclass B. We call it type A, type B, because these two classes actually occupy 53% in our total cohort. This is like the red one is metaclass A, and the metaclass B is in the steel blue. So they're differed by certain group of antigens or like self markers. So one's BCR markers. Metaclass A have a higher light chain expression and the lower CD79B expression, and also don't express IgM, not as type B, and also the HIDR expression is much higher in the metaclass A, and also there's a difference in signaling modulator like C22, 24, 44, and 72. So we just try to confirm these findings, because like again, like it's kind of in contrast to the regular or like classical believe that the FL is original, FL is looking like germinal center B. So we also perform single cell sequencing just to confirm our finding for CyTOF. So for single cell sequencing study, we pick up 6 samples, 6 FL samples, 2 from type B tumor, like a metaclass A tumor, and there's 4 from metaclass A tumor. Plus, there's the 4 react lymph node samples. So you can see the first 2 UMAP is UMAP from CyTOF data, and the third and fourth, or the second row of the UMAP is actually generated from single cell sequencing data, which is captured all the transcription, the whole transcriptome space. And here we can already see that this is germinal center B cells, and there are the 4 clusters of, so 4 type A tumors actually surrounding this germinal center B, which is even connected with germinal center B, which indicate there's a close relationship to the germinal center B cells. While on the other hand, like the other 2 tumors is very close to the memory and naive B cells. And so this is more clear depiction of like the structure of these tumors to the normal cells. And when we run this hierarchical clustering, we can also see the 4 type A tumors cluster with germinal center B, and the 2 type B tumors either activity B or memory B or plasmablast. It's quite different from the germinal center B cells. Again, like using the differential expression analysis, we can also see that some markers that we also found in CyTOF, like IgH, IgM, CD24, CD179B, and we also discovered some novel marker like 74 or TCL1A just to further differentiate them from each other. The other thing that we want to look at, like how the intratumoral heterogeneity of all these patients. So this is graph of it's kind of busy graph, but each column is 1 phenograph cluster, and each row is 1 patient. So we can see that for some patients there's multiple phenograph cluster, which means like there's multiple tumor subclones in 1 patient. And so to quantify it, we use, again, like the entropy. So the entropy measurement, we like that this is not only it capture the number of the different tumor subsets in 1 patient, but also capture like the composition. So here the example. So the low entropy, 0 entropy patient will only have 1 tumor, like nothing else, 1 tumor, 1 kind of tumor cells. And then for the patient have 2 kind of tumor cell. If you have dominant clone, let's say 90% versus 10%, you can have a little bit more entropy, but not as high as like a patient with 2 tumor subsets, which is 50-50%, 2 competing clone. And for sure, like if you have 3 different tumor subsets, and all of them are 33%, you can have much more higher entropy. So based on that measurement, we can see there's more than 30% of patients in our cohort have 0 entropy, but there's still like a fair bit of patients have very varying entropy score. And so this end up like become a very interesting factor that when we come down to the clinical outcomes. But I will talk about that later. So the last bit of result I want to share in the B cell part is also that we did the target sequencing and to assess the mutation, just to find whether there's any like mutation profile or mutations that associate with these type A or type B tumors. So we did the analysis and found that in type B, there's nothing like seems like interesting, or nothing seems to be statistically significant. But for type A, there's a couple genes like EZH2, MAF2B, and TNSF14 mutation is kind of close associated with type A tumor. That's all for the B part. Then I'm going to talk more exciting about the T parts, about T cell immune monitoring, and they are associating with the B cell population. So for the T part, we actually took a slightly different approach to look at T cells. For T cells, because they are all normals, they're just there could be residual T cell or infiltration T cell, but there's no T cell lymphoma going on. So what we do is there's a two parallel steps. So first we will do a unsupervised clustering, just try to leverage the powerful of the CyTOF like 40 marker panel to have like unbiased discovery. But at the same time, we also use the traditional approach and using manual gating to just gate out all these canonical canonical population like CD naive memory, central memory, all these Th17, Th1, all these population. And then we put the data together and use force direct map just to give us visualize that to help us navigate like different populations. So based on the red dots or like landmark dots, which is classical T cells, you will also like you will just try to use this landmark to see like what's surrounding a population. There's going to be like a population or cluster that's defined by unsupervised cluster. And then we can do some statics analysis to see the changes in different groups. So this scaffold map of infiltration T cells. So here I highlighted all these classical T cell population like Treg, EMRA, CD4, and on the that side, right hand side is CD8 population. We can see there's some like a blue bubble surrounding this T cell population, which is like defined by the unsupervised cluster. So this is the median level of all the patients. The next slide, I'm gonna just to show like when we compare the FL versus lymph node versus FL, we can like to ask question what population changed or like what the unsupervised cluster, which cluster like changed between these two groups. So the red group, the red color or red nodes are the population that like increased in the FL samples, while the green bubble or green nodes are the population decreased in the FL patients. So we can see that for compared to react lymph node on like normal situation, the increase of population, increase of T cells in FL is actually like a multi, more than one subset. There's a change in Treg, CD4, TFH, and CD8 effect memory cells. So it's really like a spectrum of T cell status changed in this FL disease setting. So there's another way we look at it is like we use heat map and also bar graph just to show like what are the population changed in FL compared to react lymph node. We can see that the whole FL group, which comprise maybe 4 or 5 different different TFH cluster, all have significant increase in the FL samples. But in contrast, for the Treg and Th1 population, there's only like 4 out of 7 cluster change in Treg. So it's really the powerful of these 40 mark panel give us just to really drill down to the very specific Treg population that changed in the F follicular lymphoma samples. So here we can see the Treg in the Treg sample, the one that changed this actually like didn't express CD40 ligand. So actually like follicular Treg cells don't really change in the follicular lymphoma setting, but the other more normal-ish Treg. 2 minutes? Oh wow, it's quick. Okay, so we also do this like T cell composition analysis just to see like holistically how whether we can use these features just to segregate the patient into different groups. So here we use 87 cluster and the composition, and we can see that there's a naive dominant group which on the far right hand side, mainly comprised of react lymph node sample. But there's also 2 groups that on the right hand side, one is TFH rich and one is Th1 rich, and then also mixed group which have mixture percentage of memory and naive B cells in the middle. This is comparison of just at the classical or like main T cell population level. Okay, this is a very interesting analysis I want to share. Like, so the next question is like whether there's any connections between T population, B population. So to really analyze this in a very granular levels, we actually borrow some idea from ecological study. So in the ecological study, if we want to study two species, whether they are co-occurred, then we so you actually what you do is you sample this co-occurrence in different sample sites. Like say one is Spiritual Park, one is North Vancouver, one is Whistler, and you see whether the two species co-present in this site, and then you can build exactly probabilistic models to predict the co-occurrence, and then you can measure like the observed co-occurrence, whether it's significant increase or decrease. So we borrow this idea. So for the geological sampling site, it's just patients. So each patient will be like a geological sampling site equivalent to that. And the T and B cell cluster is just we treat as species. And when we do this analysis, we actually got a species co-occurrence matrix, which is again very busy to look at. But we extract the information just to take the interaction from T and B, and so we plotted again in the force directed map. So it's force atlas map, and here we can see that we can actually see very interesting phenomenon that the naive normal B cells co-occur with naive T cells and central memory T cells. But there's one population which is very interesting is called a it's T10 TFH. It's one kind of TFH cell which have produced a lot of cytokines actually positively associated with type A tumor. And for the type B tumor, there's a Th1 and the CD8 EM population. Two T cell cluster is strongly associated with our type A tumor. So for clinical outcome, I just want to show that the type B tumor is related to the increased transformation risk. And also the other thing is the entropy. The tumor diversity actually contribute to the higher risk of transformation. And here we can see that the red population, they have two tumor subsets, and they actually have high entropy group compared to the green. And this is the same another example here. The conclusion, yeah. So the cytology-based immune type review extensive intertumor heterogeneity, and we define type B tumor, which is very like a novel kind of subset, which look like naive and memory B cells and have higher transformation risk. And our finding also will be informed design of conventional flow that help clinical decision making. I'll skip that. The clinical application, like we using this kind of knowledge, really like it's tied down to my current job is trying to build some automated analysis tools for LPD and acute myeloid leukemia. As acknowledgement, and that's all. So my presentation will be it's available online. So if anyone who is interested can access to the website. And our paper is also online, and the flow 155 follicular per 36 red lymph node flow data is also online. If anyone is interested, please follow the link. Thank you very much.